Hello again, and welcome to part 2 of chapter 11, which is on genetics and inheritance. So in part 1, we spoke about simple dominance. And what simple dominance says is that only one copy of the dominant allele is needed to express the trait. In the case of purple flowers, right, only the big P allele was enough, only one to have big P was enough to have purple flowers. Whether you were big P or big P little p doesn't make a difference, right? You're still purple, right? Individuals with the heterozygous genotype still had the dominant phenotype, right? So this is the case for a lot of traits in humans, but most traits are not this simple. So some simple dominant traits are like hair on your fingers. You only need one copy of the hairy finger gene, and then you're going to have hairy fingers. Dwarfism, Huntington's disease, osteogenesis imperfecta, hereditary spherocytosis. These are all diseases where having even one copy of the dominant allele is enough to show this trait, right? The only way that you would not show these traits is if you were homozygous recessive, right? Like little f, little d, little h, etc. And remember, these letters I'm choosing are totally arbitrary, right? The actual gene names have letters and numbers associated with it. So it's not really, a geneticist wouldn't use random letters. They would use the actual gene names. And that would be explained more in a genetics class. So for now, we're going to keep it simple. Just use these letters, right? And we said that dominant traits, if you have even one capital letter in your genotype, if it's simple dominance, you're going to show the dominant phenotype. Right. Recessive traits that we spoke about, we briefly mentioned phenylketonuria, right? It's a recessive trait, meaning you have the disease if you do not have a functional copy. Sickle cell anemia, little s, is recessive because you don't have a functional copy of hemoglobin. Right? Phenylketonuria is recessive because you don't have a good copy of the phenylalanine hydroxylase enzyme. Albinism, little a, is recessive, as shown here, because there's no gene for pigmentation. So again, dominant traits are usually because something is there. There's a gene that's expressing something, and even having one allele of that expression is enough for the entire organism to look just like a homozygous dominant individual. A recessive trait is a result of not having a functional gene copy, right? Not having any functional copy at all. But now we'll talk about other inheritance patterns that differ from simple dominance. So we'll specifically talk about these 5 different types of inheritance. Let's start with codominance. So we see unique traits results when 2 alleles are equally dominant together. Up until this point, we only showed that one allele is dominant over the other. But an organism might have a trait that has 2 codominant alleles. What this means is that an organism can express both dominant traits equally. One doesn't mask the expression of the other. There is no blending. Both are expressed. So for example, this flower is expressing both white and red pigment. It's not that red is completely dominant over white, because then the entire flower would be red. Let's look at a different example. In fish, the alleles for scale color are codominant, where R encodes red scales, B encodes blue scales. If a true breeding blue fish and a true breeding red fish mate, what will the F1 fish look like? Because you're told that they're codominant, right? This is unlike a Punnett square that we've done before, right? So what are the genotypes of these 2 fish? We know that they're purebred, or true breeding, so they have to be homozygous. This must be BB. This must be RR. When they have a baby fish, that fish will be a patchwork of both blue and red. So the F1 will be BR, a patchwork fish. Again, if we said this problem differently, let's say I said that R is dominant over blue, and we do this mating, then what would you expect the F1 to look like? Right? The offspring would then be red, if red were completely dominant. But in codominance, both traits are expressed equally. Right? Both dominant traits are expressed equally. So let's do one more problem. A black and a white fur panda bear are mated, and black and white fur are codominant. What are the F1 phenotypes? Right? Will they be gray? Will there be 3 whites to 1 black? Will it be all black, 50-50? No. It'll be 100% black and white panda bears. Right? This is codominant. They're both expressed together. That's what co means. The most important example that we'll talk about in humans is blood type. So blood type is codominant in humans and in mammals. So blood type refers to the type of glycoprotein found on the surface of your red blood cells. A glycoprotein is like a protein with attached sugars. And the sugars could be either A or B. They're 2 different, we'll call them glycoproteins, or antigens is the other word. These are antigens, they're proteins that are located on the surface of all of your red blood cells. If you are blood type A, that means every single one of your red blood cells has the A antigen on it, meaning the A glycoprotein is covering all of your red blood cells. If you are type B, that means the B antigen is on the surface of all of your red blood cells. Type A and B are both dominant, right? One does not mask the other. Therefore, type AB would have both A and B expressed. If A were dominant over B, then a heterozygote would only have the A antigen. But since A and B are both dominant, right, there's codominance, type AB would have both the A glycoprotein and the B glycoprotein. Type O is recessive. There's no antigen at all. And again, A and B are different antigens, meaning the immune system reacts to that. And this is why it's really important to know your blood type, because if you're type A, you could only receive blood from type A or type O. So type O is the universal donor. So why do you think that is? So what happens is, if I'm type A, my body will produce antibodies against type B blood. So if I were to put type B or AB blood into my type A body, I would have a terrible immune reaction, terrible response. O doesn't have any antigens on it. So I could put that in my type A blood, and my immune system has nothing to attack. There's nothing on this red blood cell at all. It's just a blank red blood cell, type O. So that's why type O is the universal donor. Right? Type O can be given to anybody because the immune system won't react to an empty blood cell. What type do you think is the universal recipient? So what type can receive blood from any other type? Right? That would be type AB. So AB can receive B blood, A blood, or type O blood because it already has all of those antigens. So I'm going to take a second. I want you to just be familiar with blood typing in this video right here.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Okay, so that was an example of codominance. Let's look at a different type of inheritance pattern called incomplete dominance. So some alleles are incompletely dominant, meaning neither trait can fully mask the other. So incomplete dominance gives us a blending of traits. We're going to see like an intermediate phenotype in the offspring of the two parents. So there are a lot of examples of incomplete dominance. So flower color is usually incompletely dominant. Hair texture is usually a result of many genes working together, as well as male voice tone is also a result of the blending of traits. So as an example, let's say, this is completely theoretical, you could get a very deep voice allele and a very high voice allele, and neither is dominant over the other. You would just have a middle tone voice, right? There's a blending of traits. So let's look at an example of flower color in snapdragons. So here we see two pure breeding flowers. One is RR, big R, and that's red. One is little r, and that is white. And you are told that flower color is controlled by the R allele, and it's incompletely dominant. What do you think the F1 will look like? Right, if you did the Punnett square, you would know that all the F1 genotypes would be big R, little r. And in incomplete dominance, big R, little r is pink, right? It's a blending of the two traits. So again, this is not like simple dominance. If R, big R, were completely dominant to little r, what would the F1 look like then? Right, you would get all red flowers if it were just simple dominance. Right? If it were codominance, what would you see? Right? You would see white and red patches on all the flowers. So there's an example of incomplete dominance in humans called FH. So familial hypercholesterolemia. So it's a genetic incidence of high cholesterol, and there are different levels that you can get. If you're a homozygote for the mutant allele, meaning you're homozygote recessive, that means you have heart attacks during childhood. That's the most severe form, to be homozygous recessive. And we would call those FH minus / FH minus, meaning they're mutant for both genes, and we put the slash to denote on each chromosome. So on both pairs of homologous chromosomes, there is a mutant FH gene. Heterozygotes may suffer heart attacks during early adulthood, so it's not as bad as a homozygote recessive, but they still have some kind of symptom, whereas homozygotes do not have any disorder, right? So hopefully most of you do not have this cholesterol issue, so all of us are wild type, and we do not have the disorder. But again, remember that it's not a simple dominance case. So like if your mom gave you the mutant FH and your dad was perfectly healthy, you're still not perfectly healthy, right? You're still going to have heart attacks during early adulthood because it's incompletely dominant. So it's a blending. And here's an interesting analogy to understand dominance relationships. So we know there are two alleles for every gene. We're going to look at alleles as light bulbs in this example. So if both light bulbs are on, we have to first ask, is more than one color of light possible? If no, if there's only one option and both light bulbs are on, we would say that's homozygous dominant. If more than one color of light is possible and both lights are on, we would call that codominant. Right? That's like A and B are both expressed. If only one bulb works, then we have to see, is the absence of one bulb noticeable? Meaning, if there's no noticeable difference between having a bulb on or off, we know that that's simple dominance. One allele is completely dominant, right? It gives us the same exact phenotype as the homozygous dominant individual, right? But if only one bulb works and there is a difference, right? If there's a different phenotype from the homozygous dominant individual, we would call that incomplete dominance, right? It's a blending in between the homozygous dominant and then the homozygous recessive genotype, which would be just two broken light bulbs. And so hopefully this helps clarify some of the different dominance relationships we discussed so far. So finally, there's this idea of incomplete penetrance. So some alleles that are dominant are not completely penetrant. What this means is that just because you have the dominant allele, it doesn't mean you're going to definitely express the dominant phenotype. So in polydactyly, not all individuals who inherit the dominant polydactyly allele will exhibit the trait. So even if you have this allele, and polydactyly means extra digits on the hands and feet, like this. So even if you do have the polydactyly allele, it's not always penetrant, meaning it's not going to always show. So this is incomplete penetrance. So in snapdragons, flower color is controlled by the R allele and is incompletely dominant. If two heterozygotes mate, what percentage of the offspring will be pink? So pause here and try to do this Punnett square in your notes. I'll do it here. So if we have two heterozygotes, those would be pink. So it's big R, little r, big R, little r. We're going to have big R, little r, big R, little r, and little r in our Punnett square. What percentage will be pink? 50%. Right? 50% will be pink. In petunias, flower color is controlled by the R allele and is codominant. If two heterozygotes mate, what would their offspring resemble? So again, we could look at the same kind of Punnett square, but remember that this time both alleles are codominant. So in this case, what would big R, little r represent? Right? This would be both red and white if they're codominant. So what is the answer to this question? Right, the answer is B. The red would be big R, the white would be little r. The heterozygotes would be red and white together. If flower color is controlled by the R allele and is completely dominant to little r, what would you see if two heterozygotes mate? So what would you get from two heterozygotes? So this should be very familiar. This is where we see the 3-to-1 ratio. 75% would be red, 25% would be white, if it's complete dominance. if two parents with mild FH have a son, what is the likelihood of him being completely healthy? So if two parents with mild FH have a son, that means they're heterozygotes. So what is the chance of him being completely healthy? So if the two parents are heterozygotes, that means that they're carriers. So we'll say we'll have a big F, little f, big F, little f. The only way to be completely healthy is if you have big F, big F. Right? If you have big F, little f, that means you still have mild FH. If you have little f, that means you have severe FH. So unfortunately, only 25% chance of that son being healthy. So pleiotropy is when one gene can affect many unrelated traits. So just having one mutant gene can affect a lot of other parts of the body. And a really good example for this is sickle cell anemia. Sickle cell anemia is a result of a single amino acid change in a single protein in your body. So we have over 20,000 different proteins. One of the proteins we have is hemoglobin. And we need hemoglobin to transport oxygen throughout our body and to pick up any carbon dioxide so we can then exhale it. The structure of hemoglobin is really important for it to function properly. And what happens in sickle cell anemia is that there's a mutation in the protein that causes it to fold in an improper way, and it does not function properly. When hemoglobin doesn't function properly and the red blood cells become misshapen, they start sticking together. So sickle cell anemia, again, a result of just one amino acid in one protein being switched, causes all of your red blood cells to clump together, and that can lead to blindness, liver failure, heart attack, not only anemia, right? So just one gene, one little mutation can lead to several unrelated traits. That's pleiotropy. Another example is called Marfan Syndrome. There's just one gene called FBN1 on chromosome 15 that gets mutated, and that causes all these different defects. You have very disproportionately long arms and legs. The heart is damaged. You can have aneurysms, poor eyesight, collapsed lungs, hernias and stretch marks in skin. So you have a lot, just because of one gene is mutated, you have a whole sort of different consequences. That's pleiotropy. One gene leads to many different effects. So there's this idea of epistasis. We're not going to talk too much about epistasis. I just want to introduce it. But this says that products of different genes can interact with each other. And epistasis occurs when one gene product affects the expression of another gene. Let me give you an example. Male baldness is controlled by genes. Hair color is also controlled by genes. But these two genes have something to do with each other, right? Because even if I have the gene for blonde hair or the gene for red hair, if I also have the gene for baldness, it will mask the expression of the hair color gene, right? Because if I have the gene for baldness, I'm bald regardless. We can therefore say the baldness gene is epistatic to the hair color gene. Epistatic means masks. So the baldness gene masks the hair color gene. So whether you have blonde hair gene or red hair gene, it doesn't matter. If you have the baldness gene, you're bald, right? That takes priority. The baldness gene masks whatever other genes are there. So now we could talk about polygenic inheritance, where many genes leads to one trait. So this is the opposite of pleiotropy. In pleiotropy, we said one mutation leads to many different traits. In polygenic inheritance, we're talking about how many different genes influence a single trait. Right? Polygenic means multiple genes. So multiple genes all come together to influence things like height, weight, skin color, and intelligence. Right? These are all polygenic. Right? There is no single height gene. There is no skin color gene. Right? There is no intelligence gene. They're all influenced by many, many genes. They're also called continuous traits because it's not like a yes or no. It's not like, oh, they're either yellow or green, wrinkled or round. No, you can be a wide range of colors or weights or heights or IQs. So they continue over a long spectrum, right? And many polygenic traits are multifactorial, which means that it's not only genes that play a role. They're influenced by non-genetic factors like the environment. When I say environment, it just doesn't mean like, oh, sitting outside in nature. The environment means that things like temperature, UV light exposure, nutrition, hormones in the womb, the societal setting, your stress level, all of these things can affect multifactorial polygenic traits. A great example is like intelligence. It's not just genetically determined. It depends on your stress level. Are you eating properly? Are you in the right type of school? Do you have the right kind of teacher? Do you have resources? I mean, it's not just genes, right? It's related to the environment and upbringing. And we now know that mostly every single gene is influenced by the environment. There's nothing, well, there are very few examples of something that's 100% genetic. So let's talk about a polygenic trait polygenic trait of skin color. And I'll talk a lot more about other influences on genetics in chapter 13, which is called epigenetics. But in skin color is a really good example of polygenic inheritance. Suppose 3 genes are influenced in skin color. The reality is there are over 100 genes that influence skin color. But let's say there are only 3, A, B, and C. In the parent generation, let's say you get the lightest possible individual mating with the darkest possible individual. Right? So this person has a total of 6 capital letters. That's the darkest. This one has 0 capital letters. That's the lightest. When these 2 parents mate, the F1 generation will be an exact intermediate between both extremes. Right? They're going to have 3 capital letters each. But what will happen when the F1 generation expands? We'll see this bell curve. This is called a bell curve because it looks like a bell. And this is a normal distribution, meaning in the next generation, most of the population, or you could say it is most probable that you're going to get something in the middle that also has 3 capital letters. But it's also possible to get all 6 capital letters or 0 capital letters. So again, this is a spectrum. And this just shows that when you have multiple genes, right, you get more of this bell-shaped curve. The more genes that influence a trait, the more diversity of that trait you're going to get. So again, if you have one child in the F2, chances are it will resemble the parents, but it can also or not just as likely, but it can also be any other shade of skin tone. Right? And this is not just skin color. This is height, weight, intelligence, BMI, etc. Most traits are spectra. That brings us to part 3. So we're going to take a look at patterns of inheritance in human diseases. So when we talk about human diseases and genetics, right, we're talking about genetic disorders, meaning this is something that you get from your parents. Right? This is not like cancer, which is a result of a mutation that's usually happening during your life. Genetic disorders are caused by alleles inherited from the parents at fertilization. So you can have a disorder that's autosomal, or you can have a disorder that is linked to the sex chromosomes. So let's talk about autosomes, right? Remember, an autosome is a chromosome other than a sex chromosome. So chromosomes 1 through 22 are all autosomes. Genetic disorders caused by genes on autosomes are called autosomal disorders. So most genetic disorders are autosomal because they're on chromosomes 1 through 22, right? Chromosome pairs 1 through 22. Some genetic disorders are autosomal dominant. What this means is that this gene is on an autosome. An individual with big A has the disorder, as does somebody with big A, little a, right? Even one copy of the big letter is enough to have the disorder. Only an individual with little a will not have an autosomal dominant disorder, right? Other genetic disorders are autosomal recessive. That means an individual with big A is perfectly healthy. Individual with big A does not have the disorder but is a carrier, right? The heterozygote is a carrier because the disease allele is little a. So little a would be an affected individual with the autosomal recessive disorder. So we said genetic disorders could be either on an autosome or a sex chromosome. We're only talking about autosomes right now, and it could be autosomal dominant or autosomal recessive. Let's look at an autosomal dominant disease. You should know the example of Huntington's disease. And autosomal dominant patterns of inheritance say that if parents carry even one copy of the diseased allele, they are affected. So what this means is that two heterozygotes are capable of having a wild-type child with two copies of the recessive allele, which means that child could be healthy. So in Huntington's disease, what we see is that there's a protein that gets misfolded. And when that protein gets misfolded, it starts clumping up and damaging nerve cells. Having even one version of the clumped protein is enough to cause the same amount of damage. So you're normal if you only have the normal protein, both copies of the normal protein. If you have just one copy of the misshapen protein, you have the disease. There's no difference between having just one copy of the bad allele or two copies of the bad allele. Right? It's dominant. Having just one copy is enough to show the disorder. So Huntington's disease is an example of this. And again, it is just because of this mutated copy of a gene of a protein called Huntington. And unfortunately, there's no effective treatment. And once symptoms start, death follows within 10 to 15 years. So that's Huntington's disease, autosomal dominant pattern. Let's look at an autosomal recessive pattern of inheritance. And in this case, if both parents carry one copy of a recessive gene, they are unaffected. But they're capable of having a child with two copies of the recessive gene that is affected. Right? So you can have two carriers, right, that have one copy of the normal gene, one copy of the bad one, or the mutated one. It's possible for them to have an unaffected child, 25% chance, 50% chance of having a carrier child, and a 25% chance of having an affected child. And that's shown in this Punnett square here. So if two carrier parents have babies, there's a 25% chance that'll be normal and a 25% chance that it will also have the disease. Examples of these are a disease called methemoglobinemia, which causes the appearance of blue skin, and also cystic fibrosis. So we'll talk about cystic fibrosis. And cystic fibrosis is the most common lethal genetic disease among Caucasians in the US. About 1 in 20 Caucasians is a carrier, and about 1 in 2,000 newborns has cystic fibrosis. So it's caused by a defunct protein called CFTR. So the CFTR protein normally controls the passage of chloride ions across the cell membrane. So functional CFTR proteins make sure that chlorine is eliminated from the cell, so the inside of the cell has very little chlorine in it. So if you have at least one copy of the proper gene, that's enough to keep all the chloride out. All you need is just one copy. of the proper protein. However, if you have no functional copy of the CFTR protein on chromosome 7, then chloride ions cannot pass through at all. And what's going to happen is there's going to be a lot of thick mucus built up on the outside of the cells. So when chlorine is stuck inside, mucus starts building on the outside. And this really thick mucus actually causes pancreatic deficiencies, in addition to breathing issues and lung issues. So it can also affect the pancreas, because the pancreatic duct can't secrete the right kinds of enzymes into the small intestine. So CFTR, again, is a recessive disease because you need 2 bad copies in order to get the disorder, as opposed to Huntington's, where even 1 copy of the bad diseased allele was enough to get the disorder, because it was dominant, right? So those were autosomes. Now let's talk about sex-linked genes. We know the sex chromosomes are X and Y. And in humans, females have 2 X chromosomes, males have 1 X chromosome and 1 Y chromosome. And just by doing a simple Punnett square, right, 2 X's, an X and Y, right, male, female gametes, all eggs always have an X. In sperm, 50% have a Y, 50% have an X. So by just doing a Punnett square, you can see there's a 50% chance of an X egg and an X sperm meeting, 50% chance of a Y sperm and an X egg meeting. So that means there's a 50% chance of producing a female, 50% chance of producing a male, biologically speaking, right, as determined by an X and Y chromosome. So X-linked inheritance is sex-linked inheritance. This means that these genes are on the sex chromosome, on the X chromosome. The term X-linked is used for genes that have nothing to do with sex or gender, right? They just have to do with being on the X chromosome. Any gene that is X-linked is called sex-linked because it's just on the X chromosome. It has nothing to do with gender, right? And the Y chromosome does not carry these genes. The X-linked inheritance was discovered in the early 1900s, headed by Thomas Hunt Morgan, which is one of the most famous geneticists. He worked with fruit flies. And fruit flies are really easy to work with because they can be used very cheaply. They generate, they reproduce very quickly. You can keep lots of them in the lab. Very cheap, easy to find. And what he saw, without going into too much detail, normally he took, so when we see, you took a white-eyed fly and a red-eyed fly. And again, the F1 we would expect to see only red-eyed individuals, right? Because red would be dominant. But what he saw in the F2 was something really weird. He saw that all the females were red-eyed, and half of the males were red-eyed, and half of the males were white-eyed. So this was unique. This was unlike any kind of Punnett square that Mendel had done. And he finally realized that the gene or the allele for red eyes must have been on the X chromosome. So it is more likely that the females, right, they're going to always have at least 1 X with the red eyes. So all the females will have red eyes. But with the males, if they got the recessive allele and a Y, they would be white-eyed. So again, this does not show the 3-to-1 ratio that we would have expected if it were on an autosome. This leads to X-linked inheritance. Right? When you see a deviation in the 3-to-1 ratio, it must be on the X chromosome. So X-linked recessive disorders affect more males than females. Why is that? Right? Males only need to inherit 1 X-linked recessive allele to express the recessive disorder. Remember, males only have 1 X chromosome. If they happen to get a bad X chromosome, they will definitely have every recessive disease. Females must get 2 bad X chromosomes to express the recessive disorder. Right? And that's less likely to occur. So what we see is X-linked recessive disorders are more common in males. Hemophilia is a good example of an X-linked recessive disorder. And hemophilia is a disease that results in blood that doesn't clot, because the clotting factor genes are on the X chromosome. If a healthy carrier mother and a healthy father have a child, what is the likelihood of having a daughter with hemophilia? What about having a son with hemophilia? So this is a tough kind of problem. Let's try to figure this out using a Punnett square. So the way we show Punnett squares with sex-linked disorders is we just show the sex chromosomes. So a healthy carrier mother, right? Let's say big H is healthy, little h is hemophilia. This mother is a carrier, so we're going to say she has one X chromosome that's healthy, she has one X chromosome that's unhealthy. A healthy father is XY. What does the father have, a big H or a little h? Right, he's have a big H because he's healthy. So this is our Punnett square now. So what you'd have to figure out is what are the chances of getting a daughter with hemophilia, right? And what is the chance of getting a son with hemophilia? So in this case, we would have, so you should be doing this in your notes. So what is this genotype and phenotype? This would be a healthy female. What about this genotype and phenotype? This would be a healthy female as well, because there's still one copy of the healthy gene. What would be the phenotype and genotype of this individual here? This is a healthy male. And finally, what is the genotype of this individual here? This is an affected male. So going back to the problem, it says, what is the likelihood of having a daughter with hemophilia? The answer is 0%. What about a son with hemophilia? There's a 25% chance of getting a son with hemophilia. I just want to point your attention to the way the wording is in the question. Because suppose the question said, a healthy carrier mother and healthy father have a child. If they have a son, what is the likelihood of him having hemophilia? All right, that's a different question. All right, that's saying if you have a son, if you're only looking over here, what is the likelihood of him having hemophilia? And in that case, it would be 50%. Right? So just make sure you're reading the question properly. And here is the Punnett square that I just drew out. Right, so this is how we draw the genotypes for sex-linked traits. We just draw the X and the Y with the letter as a superscript. Hemophilia is an X-linked recessive disorder. If an affected female and an unaffected male have a boy, what is the chance that he will have hemophilia? So let's do this problem out. Pause it here and do it yourself. So affected female is XH, X little h. Unaffected male, we have big H and Y. If they have a boy, what is the chance that he will be a hemophiliac? Well, unfortunately, 100% chance. Right, or 1. Because the boys only get an X chromosome from mom. If both of mom's X chromosomes have hemophilia, the son is going to get hemophilia. So this explains why males are more impacted by X-linked recessive disorders. Another example is color blindness. So red-green color blindness is on the X chromosome. Muscular dystrophy is caused by the absence of a muscle protein that's on the X chromosome. Adrenoleukodystrophy is another X-linked recessive disorder that results in the peroxisomes not functioning properly. And of course, hemophilia. So here's a table of some X-linked recessive disorders and also some X-linked dominant disorders. So finally, we're going to talk about pedigrees. So a pedigree can be used to study inheritance because it can help us diagram all of a family's relevant genetic traits. And we can see how those traits get passed on from generation to generation. In a pedigree, we like to include as many generations as possible, at least both sets of grandparents of an affected person. And we can use Mendel's laws to help analyze if a trait is being determined by a single gene, is it dominant or recessive, etc. So pedigrees are very, very useful to studying inheritance. If you're interested in pedigrees and genetic inheritance, you might want to pursue a career as a genetic counselor. What a genetic counselor does is look at pedigrees of different families and will figure out what the likelihood is of a couple having a child with a genetic disease. So genetic counseling is an up-and-coming field. It is very lucrative, so you make a really nice salary with it, and you're helping people by using genetics and counseling together. So here are some symbols that we use in pedigree analysis. Males are squares. Females are circles. A diamond is sex unspecified. And if they're unfilled shapes, those are healthy. If they are filled, that means they have the disease. If you see an X, that means they're dead. This is if a brother and sister mate. We're not going to see that. And we're not going to see this too much if you have multiple progeny. What we see is there are Roman numerals that define each generation. So these are the parents. This is an affected father mating. This is a mating line, mating with an unaffected mother. This is the line of descent, meaning they have these two children. They have two boys, one that has the disease, one that does not have the disease. We number each generation in Roman numerals, so generation 1 are the parent, generation 2 is the F1, and each individual has a number, like 1 or 2, associated with each individual within a generation. So pause here and make sure you're familiar with these pedigree symbols. This is very important. So now I'm going to tell you how to recognize dominant traits in a pedigree. There are 3 key aspects of pedigrees with dominant traits. An affected child will always have at least one affected parent. Right? Anytime you have a child with a disease, the father or mother will also have it. Right? It's the only way that a dominant trait can be passed down to a child is if the parent has it too. As a result, we see vertical patterns of inheritance. That means it gets passed down one generation to the next, top to bottom. Right? And if it's a dominant trait, that means 2 affected parents can possibly produce an unaffected child if both parents are heterozygotes. Right? So there's a 25% chance of having an unaffected child if 2 affected parents are carriers of a dominant disease. So this is, again, an example of Huntington's disease, showing a vertical pattern of inheritance, which shows the dominant inheritance. Right? Every affected person has at least one affected parent. That's how you know a dominant pedigree. So here, this is from the book, this is showing 2 heterozygotes are both affected by the disorder, but it's possible for them to have an unaffected child as well. Right? And here you can see that a heterozygote is affected and can pass on that dominant allele to the next generation. And both males and females are affected at equal frequency. So make sure you're able to identify an autosomal dominant pedigree if you're given one. There are different aspects to recognize with a recessive pedigree. So recessive traits show a horizontal pattern of inheritance, meaning siblings can share the recessive trait, even though their grandparents do not, or their parents do not. So you'll see individuals within the same generation have the trait, whereas it can skip generations. Affected individuals can have two unaffected parents, right? So it's possible that two seemingly healthy parents have an affected child that has a recessive disease. And all of the children of two affected parents will also be affected, right? If it's a recessive disease. Right? So we see it's possible that two unaffected parents can still have an affected child. Right? Here, two unaffected parents have affected children. Right? Because these must be carriers for the disease. This is like cystic fibrosis. So here we can see there's a skipping of generations, right? It skips generations, then it reappears, right? Most affected children have unaffected parents in a recessive pedigree. Right? And both males and females are affected equally. This pedigree tracks an autosomal dominant disorder. What is the phenotype of individual 1, 2? Right? That is B. It's an affected male. What is the genotype of 1, 2? So we know this person must be heterozygous, because if it were homozygous dominant, that means all of the children would have been affected. But we see here, out of 5 children, only 3 were affected, which means the person must have been heterozygous. The Blue People of Troublesome Creek is a very interesting case of a recessive disorder. It's been documented for centuries, but it wasn't until recently that we knew what caused it. So methemoglobinemia is a genetic disorder. It's relatively harmless. It just results in the accumulation of a certain type of hemoglobin in the blood that looks more blue. So enzyme tests indicated that the result comes from, or this disease comes from, a lack of an enzyme called diaphorase. So when you don't have any copy of chromosome 22, of diaphorase on chromosome 22, that means you have this blue skin. What type of inheritance pattern is this? So this is showing the Fugate family that lived in Kentucky that had 7 children and 4 of them were blue. And they saw the blue appear 2 generations later. So what kind of disease is this? It must be recessive, right? It must be a recessive type of disorder, right? Because they lost the function of this diaphorase enzyme that converts the blue hemoglobin into the red hemoglobin. So like I mentioned before, hemophilia is an X-linked recessive disorder that shows us different patterns of inheritance. So if you looked at a pedigree of hemophilia, you would see something very different. You would see more males than females are affected. It's possible that an affected son has 2 normal parents, right? In order for a female to have the disease, the father must have it, right? And it usually skips a generation from the grandfather to the grandson. So this is a generation that was skipped, right? And if the woman has the characteristic, that means all of the sons will have it because she passes on her X chromosome to the sons. So this is a very famous X-linked recessive pedigree that shows hemophilia in the royal families of Europe. So Queen Victoria was the first carrier female of hemophilia, and eventually it was spread throughout the royal families of Europe. Right? Since it's recessive, and again, what the royal families used to do was intermarry. They used to keep on marrying each other. So that increases the likelihood of 2 recessive alleles coming together. Right? So this is not very good. So what happens is you see that throughout the royal families, you see more and more hemophilia. And that all came from Queen Victoria at first. Right? So out of her 26 grandchildren, 4 grandsons had hemophilia, and 4 granddaughters were carriers. So what type of inheritance pattern is shown below? This must be autosomal dominant. Right? Males and females are equally affected, does not skip a generation. Every affected individual has an affected parent. That is the end of chapter 11. So let's just recap everything we spoke about so far. We said cells could be haploid or diploid. Haploid cells have 1 set of chromosomes. Diploid cells have 2 sets of chromosomes. Chromosomes can be autosomes, right? Pairs 1 through 22, or sex chromosomes, which can be X or Y. Chromosomes contain genes that have alternate versions called alleles. Each allele could be either dominant or recessive. If there are 2 identical alleles, we say that it's a homozygous genotype. If there are 2 different alleles, we say that it's a heterozygous genotype. And genotype plus the environment influence phenotype. So that is the end of our genetics chapter. So please review this, this part and part 1 over and over again. Do as many practice problems as possible. There is an interactive assignment on Connect that will have you work through several different problems. Also in the lab, this week's lab will be practicing a lot of Punnett square problems. It is essential to practice, 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 practice. You can watch this over 1,000 times, but you're not going to understand it until you actually draw Punnett squares in your notes. So please take a look at the lab, do practice problems in the back of the book. Of course, on the lecture review, you're going to have some practice problems. So just make sure to practice, because that is the key with genetics, is just practicing. So you can also have a test on page 206 in the book. You can look at some questions as well that show you the answers. So that is it, and I will see you next time when we discuss the central dogma of biology in chapter 12.